barrier and the speciation event have to be roughly the same age. But the most important thing about it is that the barrier cannot be younger than the species. The barrier is younger than the species. Try something else. Mole rats in Tibet. Aren't they cute? Well, probably to their mothers they're cute. So here are the phylogenetic data on four species of Tibetan zoophores. Here's the geographical data. This is fairly complicated. Well, here's A. All right, A is here, and it's isolated. Here's this nice big mountain range that separates A from everybody else. So that looks like that could be the reason for this split, this speciation event here, that produced this and those species. And the species B is over here, and its distribution is close to that, but it's not overlapping. They're actually disjunct here. And B plus CD has to do with the ancestral distribution, the geology, geography of that ancestor and this speciation. So B versus those. And what you have is a major river here and a nice disjunction there, a drainage pattern disjunction there. And then you have C and D. D is here, C is here, and here, oh look, you have another major river system. So you have one old major mountain building event and the evolution of two distinct drainage patterns that provide barriers. And we know the ages of each of those. If you have a molecular clock hypothesis or fossil data that you can depend on, that you feel are fairly robust, then the ages of these species should correlate with the ages of these physical alterations of the landscape. So there's one. There's another. And there's another. So the Yellow River drainage here on this side of the mountain, like that. And then these turn out to be, this and this actually turn out to be correlated with glaciation periods which ended with an alteration of the river drainage patterns. And the ages of these turn out to be highly correlated with the molecular clock estimates for the ages of the species. Oops. And oh look, this is very convenient. This is older than that, and this is older than that. And these ages turn out to be very close to the ages of the geological events. That's how you go about documenting vicariant speciation. Now, what happens if the geography doesn't change? Suppose those geographic barriers are already in place. Is it still possible to have allopatric speciation? Well, yes, it is. The difference is that in order to have allopatric speciation, when the barrier is already there, you must be a member of a species, an ancestral species, that's capable of getting across the barrier. So you have to be able to fly over or swim across the barrier. Now, it works best in cases like this. It works best if you're not able to cross the barrier easily. So if it's a rare event, if only a few members of your species are capable of getting across the barrier, and for some reason it's even more difficult to go back, then it's possible that you can have a rare dispersal event which establishes a small founder population. which isolates itself as a result of the dispersal from its ancestor across a pre-existing barrier. It isolates itself from its ancestor and becomes, over time, becomes a different species. Now that's a situation in which what we call active allopatry speciation. The species itself did something actively to isolate part of itself from the ancestor. And that's sometimes called peripheral isolate speciation or active allopatry speciation. So a dispersal event in the Andes, for example, we have a lot of cases of this because you have these situations, you have periods of time when there's a warming period and mountain low passes in the high Andes open up. And for a brief period of time, it's possible for things to disperse through that. Then it gets cold again, and that pass closes off. So you may have a break in the barrier for a short period of time caused by climate change or something like that. Or you may actually have a rare event. Now, one of the ways, for example, one of the ways that we know that the current story about the way species got to the Galapagos, all the evolution on the Galapagos, we know that that's a complete fabrication because we have more geological evidence now and we know something about the biological capabilities of the organisms. For example, in the Amazon, there is a terrestrial tortoise that's the sister species of the giant tortoises on the Galapagos. So how did those big turtles get out to the Galapagos? Okay. Well, right now the current thinking is that they got on top of a bunch of trees that had fallen down during a big flood. And they were carried to the ocean. And then they just drifted out to the Galapagos. Okay, well, one way to test that hypothesis is to go and see how often you find logs floating down the Amazon or down the Magdalena or something with turtles on them. Another would be to pick up one of those turtles, walk over to a river, drop it in the river, and see what happens. Because it's possible, there was even a hypothesis that they hyperinflate their lungs and they floated out to the Galapagos. Okay, so what happens if you go to the Amazon, you find one of these turtles, you walk over to the river and you drop that turtle in the river. What does it do? Does anybody know? Sinks to the bottom of the river and then walks across the bottom of the river and comes out on the other side. Okay, that's how they cross bodies of water. Okay. That did not happen going from South America to the Galapagos. So, once upon a time, like 12 million years ago, the floating hotspot that produced the Galapagos was very close to the western coast of Costa Rica. And the sister species of the iguanas that live on the Galapagos now lives in Costa Rica. And 12 million years ago, there was an island close to Costa Rica. And it turns out that the molecular clock estimates for the age of divergence between those lizards is 12 million years ago. So the lizards, the iguanas, didn't swim out to the Galapagos either. 12 million years ago, their ancestor walked out to the island. And then over the next 12 million years, plate tectonics moved them out into the ocean. So sometimes there's long distance dispersal, sometimes there isn't. In this particular case, an option opened up, a few things dispersed, and then they became isolated. And in isolation, they became different species. Now, the interesting thing about this is that on a phylogenetic tree, we would give this a name, and we would give this a name, and it would appear that they were distinct species, both the species distinct from the common ancestor. Not necessarily true. This may still be, this is a case in which we would say that the ancestral species survived the speciation event. In vicariance, the assumption is that the ancestral species is completely subdivided and disappears into the two descendants. But in this particular case, the two species genetically may be so asymmetrical that, in fact, we could say that the ancestors still surviving. We have difficulties in recognizing ancestors in a practical sense, but that is one potential outcome. The ancestral species either became B and C, or this species C is actually the same as the ancestor A. Okay. Active dispersal. Number one, pre-existing barrier. The descendant species, the species that dispersed, must be younger. The speciation event must be younger than the barrier. Or to put it the other way, the barrier has to be older than the speciation. Now, what we call these things, these isolated populations, when we say that they are just allopatric populations of one species, or when we say this is a different species, depends on a number of things. It depends a lot on time. It depends on how long the barrier stays open. It depends on whether or not there is any back colonization, whether or not there is continued gene flow after the dispersal event, and it depends on time. So we may, in fact, have a situation in which it's ambiguous. We may have caught speciation in the middle of the process. So if you can continue to have a lot of dispersal, if that opening remains, so that the dispersal event actually allows a lot of members of the population to disperse, you can end up with a situation that would look very much like vicariance. It wouldn't be obvious. It's also possible that you have range expansion or changes in geographic distributions after the speciation event that make the geographic distributions of sister species look like something else. That's one of the reasons that you have to have independent timing of the barrier, independent dating of the barrier. So just because, for example, just because you have two sister species that appear to have ranges that are about the same size, it doesn't mean that it was vicariance. It still could have been peripheral isolate speciation. So now let's talk about South American cats. Okay, so here are two little cats from down in this part of the world, one of which is fairly widespread east of the Andes, one of which has a very limited distribution west of the Andes. And they are younger than the Andes, younger than that part of the Andes. This part of the Andes is really old. This part of the Andes is Cretaceous in age. And those cats are not 80 million years old. So this looks like a really nice case of peripheral isolate speciation from the east to the west. The species are younger than the barrier. Dispersal corridors, as most of you know, have opened and closed multiple times in the history of the Andes as we have warming and cooling trends. So here's how you distinguish between the two different allopatric modes of speciation. It's complicated, but you can do it. Now that's just how is speciation started. The other part of speciation is how is it completed. And there are a number of ways that speciation can be completed. One is just pure drift. Get separate, get different. If you don't communicate with each other for a long time, you end up not being able to talk to each other. Founder effect is another way. Genetic bottleneck is another way that speciation can be completed. The genetic subset of the population that establishes a new species is such a small percentage of the total ancestral genome that there is very rapid differentiation and divergence. Or you can have disruptive selection, is directional selection of two different vectors in the two different populations. So there are a lot of ways that speciation can be completed once you establish geographic separation. But the phylogenetic patterns and geographic distributions themselves don't tell you anything about that. For that, you need to do genetic work and population work. Okay, now it's not really so simple as to say if you have asymmetrical sized geographic ranges for sister species, it's peripheral isolate speciation, and if they're more or less the same size, it's vicariant. Because you can have real vicariant speciation as passive allopatric speciation that subdivides an ancestral population, ancestral species, asymmetrically. And that's sometimes called microvicariant. So you have a little barrier here, and you end up with sort of carving off a small percentage of the population. In that particular case, you end up with something that looks like peripheral isolate speciation. And even though it's vicarious, you still have the same dynamics of founder effect, small population size, that can come into play for completing speciation. Also, allopatric speciation does not always produce nice bifurcating speciation events. Okay, rapid episodes of microvicariance or peripheral isolate speciation can produce a situation in which the actual phylogenetic relationships look like that. This is a hard polytomy. This is a situation in which you have a burst of speciation events that are all more or less contemporaneous and generally involve speciation modes in which the ancestral species survives the speciation events. When you see something like that, if it's a hard polytomy, you should be suspicious that one of those surviving species might actually be the ancestor of the others. It's difficult to document, it's difficult to figure out, but that's a possibility. So, now, non-allopatric speciation modes. There's a huge range of these things, and they all are based on how much geographic overlap there is between the new species that are produced. Okay, but they all refer to modes of speciation in which the species subdivides itself without complete geographic separation. They are also called divergence with gene flow modes of speciation. One of them, John Endler's favorite mode of speciation, is called parapatric speciation. The idea is that you have some sort of environmental gradient, and the species population is aligned along that gradient. And at the ends of the gradient, what you have are, let's say this is temperature. And so you have a species that's all along this temperature gradient. And over time, you end up with a species in which you have low temperature adapted members of the species. Here you have high temperature adapted members of the species. And as time goes along, eventually what you're going to have is two different species form. What keeps them from dividing into two species is that here in the middle, everybody's still reproducing with each other. The cold adapted, the warm adapted members of the species are still reproducing with each other. Now, if it turns out that these quote-unquote hybrids between the two extremes, if they are at a selective disadvantage relative to the ones at the extreme, then you can have this very interesting situation in which hybridization can continue to occur, interbreeding can continue to occur here, but none of the information in the hybridization events goes out. So you have warm adapted organisms breeding with cold adapted organisms here, but none of the genetic information about being warm adapted gets up here. None of the information about being cold adapted gets down there, because the organisms that carry both kinds of information are at a selective disadvantage or are at a reproductive disadvantage. And so you have hybridization going on, but the existence of a hybrid population here that is not communicating with either side in the second generation actually creates a reproductive barrier between the extremes. And you can end up with two species being formed even though gene flow has been continuous throughout the process. Now that's very cool. I mean, it's a really cool mechanism. John Emler wrote a whole book about it. It's very cool. There's only one problem with that. Every example we listed in the book, we have no phylogenies. We don't know if the actual parapatrically distributed hybridizing species are actually each other's closest relatives. If they're not sister species, then they're just two different species from two different speciation events that happen to be together geographically and happen to be compatible enough that they can make mating mistakes. And the current hybridization and geographic distribution has nothing to do with how the two species were formed in the first place. And unfortunately, every single example in John Emler's book is undocumented with respect to phylogenetic relationships. So we don't know. Okay, here's the whole deal. You get something that looks like that. Sister species that have distributions like that, and you have hybridization, you have gene flow occurring within there throughout the process. And then these are the factors that influence whether or not parapatric speciation is possible. Here's the contact zone. Small number of individuals in the contact zone that are at some kind of disadvantage with respect to the parental types. Now here's an example that might be parapatric speciation, having to do with these protists, foraminifera. All right. So here are what have been called two types, two species. Now we recognize two types of foraminifera. Now they're phylogenetically distinct. They might be species. Now let's find out what's going on. All right. They might be different species, and they're sister groups. So we know that if they are different species, they're the product of a unique speciation event. Now it turns out that blah, 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 blah. The short answer is that they live at different depths. Okay, basically there's one shallow water species and one deeper water species. But there's a lot of overlap in the middle, and they reproduce in the middle. But the interesting thing is that the hybrids in the middle, if they move up into shallow water, they don't survive. If they move into deeper water, they don't survive. So you have, right through here, you have this little hybrid zone. But they don't contribute back to the parental species. So there's no net communication across the species. So there's an environmental climb, which in this case is temperature and light and water pressure. Now we don't know anything about selection and gene flow in these things other than the fact that we can pick up, with molecular data, we can pick up hybrids. Now there are other cases where we actually know something about the situation. We may know that there's hybridization going on. We may know that here's one species and here's another. They're parapatrically distributed there in Costa Rica. There's overlap. There's hybridization. So let's go study parapatric speciation. They're not sister species. They're not each other's closest relatives. They're just two different species that happen to overlap geographically. And because they're members of the same genus, from time to time when it's really dark out there and in the jungle and you can't really see all this nice bright coloration, mating mistakes are made. And everybody has a good time. So This is not enough, just seeing that kind of distribution pattern. They have to be sister species. You would have to find something like this. If they're sister species, then you could consider the possibility of parapatric speciation. Sympatric speciation. This is the most interesting in some ways and the most controversial mode of speciation. This refers to a situation in which a new species, descendant species, emerges completely within the geographic distribution of the ancestor. So there's no climb, there's no gradient involved. And there are a number of, so these are speciation events by extreme disruptive selection. And one of the most commonly mentioned mechanisms are things that have to do with sexual selection and the production of dimorphic species and variants on dimorphic species and things like that. So in this particular case, you have, in parapatric speciation, speciation goes to completion even in the face of some gene flow, but there's some geographic spread. In sympatric speciation, there's no geographic spread, but gene flow is immediately cut off. So we have this relationship between geographic distance and gene and genetic communication. They are members of each population, encounter each other on a regular basis, but they don't reproduce. Irrefutable evidence of sympatric speciation in natural populations remains elusive. So in 2012, very interesting kind of speciation. We don't have any examples of it. Same thing with parapatric speciation. Very interesting mode of speciation. We don't yet have any cases of parapatrically distributed sister species. And you have to show that they are sister species that are distributed sympatrically. And they have to be reproductively isolated, which means for sympatric speciation, this is the one kind of speciation event for which the biological species concept is really critical. Guess which kind of speciation mode Ernst Mayr was 100% opposed to during his entire career. The only mode of speciation that would actually have demanded his species concept. Oh well. And you also have to show panmixis within the two sympatrically distributed species. So they cannot be reproducing with between each other, and they have to be broadly panmictic within. They have to be reproducing widely within each one. They also have to differ from each other. There has to be at least one apomorphic trait that is directly associated with the inability to interbreed. Whether that's a trait that has to do with sexual dimorphism or courtship behavior, or whether it is a size difference or a habitat preference difference, anything, but it has to be an apomorphic trait in at least one of the species that's associated with we can't reproduce together. Okay. And you have to be able to show that they never have been allopatric in the past. They are sympatric now, and they have always been sympatric. That can be difficult to document. But see, now you understand why it's really difficult to document sympatric speciation. So you have all of those things have to be true for a group. But there's one possibility that may actually be true. On a little tiny island in the Pacific Ocean, there are two species of palm tree, the thatch palm and the curly palm. Are they sympatrically distributed? And in the publication they said, yes, they're sympatrically distributed, by which they mean that in 20% of the sites they sampled on the island, the two trees occur together. Okay, well, all right, maybe that means they're sympatric, maybe it isn't. Are they sister species? Okay? Yes and no. Yes, they're sister species, but one of the species appears to be more than one species. Or, if this is actually one species, maybe they're actually all the same species. Or maybe one is the ancestor and the other is the descendant. So, yes, kind of. The problem is, remember what we said about having a robust tree? Now, it's really difficult to get almost 3,000 equally parsimonious trees for a 3-taxon statement. But that's one of the wonderful things about genetic data, sequence data. DNA sequences are so ambiguous that you can get an enormous amount of complexity for almost no investment of money. The reason for that is because at any one site in any DNA sequence, after the third mutation event, the probability that the next mutation will produce homoplasy is 100%. Okay, so the thatch palm is not monophyletic, and the tree isn't necessarily very robust. However, soldiering on, and they may or may not really be truly sympatric. So yes, they are sort of maybe sister species, and yes, they are sort of maybe sympatric. And so then the next thing is, are they reproductively isolated? There they are living together. Yes. Okay. All right. We have this one thing. They flower at different times. So they're reproductively isolated in terms of their flowering behavior. Is there panmixis within each of these different species? Yes. And interestingly enough, they're both wind pollinated, which means that if they were capable of interbreeding, they would, because the wind would just sort of pollinate everybody. So that really, the genetic data and their behavior, their pollination behavior says, yes, they're reproductively isolated. Could they have been allopatric in the past? Well, probably not. It's a little island. It's kind of had a fairly uneventful geological history. So there they are. Now, it's possible, you look at this, maybe at some point in time, but not really, especially considering how small the island is. It's in the middle of the ocean, and these things are wind pollinated. So, more than likely, they were not ever allopatric. So here is the scenario for producing these things. Now notice here, we have this situation in which at this point, remember these are people who are trying to say this is sympatric speciation. And then they throw this in and say a population colonized a different soil type on the island. And it's not sounding exactly like sympatric speciation anymore. But they're calling that an ecological shift rather than a geographic dispersal event. And there's a change in flowering time and the mechanism, and voila, what they believe that you have is one species that lives in basic soil, one species that lives in acidic soils, but somehow they're still supposed to be sympatric. So what happens is you end up with these papers that say, evidence of sympatric speciation in blah, 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 blah. You go through it, you go through it, you go through it, and ultimately, almost inevitably, you end up at some point finding something that looks like some kind of microgeographic event that could physically, spatially isolate the populations. And if that's the case, then maybe it's not really sympatric speciation, and that's the point at which everybody starts arguing about what is the proper definition of sympatric. So, why is it important to establish that they weren't allopatric in the past? It's because the allopatric modes of speciation require that speciation is initiated and completed when the populations are isolated. And the other modes of speciation require that the speciation is initiated and completed even when there's gene flow and geographic overlap to some degree. Now, there is a hybrid speciation mode that kind of incorporates both. That was Ernst Mayr's favorite mode of speciation. It is currently being called ecological speciation, but that's just a rebranding of an older concept. And this is what Dolph Schluter and Jonathan Losos are making lots and lots of publications about. Speciation by reinforcement is a really interesting mode of speciation. It goes something like this. You have an ancestral species which is either subdivided by vicariance or by dispersal. And then there is some isolation, there is some divergence in isolation, and then they secondarily come into contact. When they come into contact, they are still closely related enough to each other, they're still similar enough to each other. They are partly diverged, but they're similar enough to each other that in the areas where there's overlap, there's the possibility of making a mating mistake. There's the possibility of hybridization. And in those cases, now this is secondary hybridization. This is not ongoing, this is how this is different from parapatric speciation. Allopatric, secondary overlap, hybridization, and the hybrids are at a fitness disadvantage in either parent. And that hybrid disadvantage reinforces the penalty for making a mating mistake. That's why it's called reinforcement. So it reinforces the divergence. And it amplifies the characteristics that make the two species not recognize each other. So that's why those traits were originally called isolating mechanisms, reproductive isolating mechanisms. Now, this is called speciation by reinforcement or alloparapatric, or like I was saying, it's now being called ecological speciation. And it goes something like this. Allopatric speciation, they begin to differentiate. The barrier goes away. There's dispersal. They come into contact with each other. And backstory. That secondary contact, secondary contact produces two possibilities. One is that when they come back together, they just haven't diverged very much, so they just remix. So you start with one ancestral population, two allopatric populations, they come back together and it's one ancestral population again. There's no net speciation. And in cases like that, we would never know about it. However, it's possible that that secondary contact will reinforce and complete the speciation process. Because you have some individuals from one group breed with members of the other group. But there are reproductive incompatibilities that occur. In other words, they reproduce, they copulate, but things don't go well. And these are called post-zygotic isolating mechanisms. Genetic infertility, incompatibility. They mate, but it doesn't work. Now, this is very costly, this to individuals. You've wasted all your time reproducing or copulating with somebody, and you're never going to get any reproductive benefit out of it. So selection will favor organisms that don't make those mistakes. Or have a situation in which you have the evolution of assortative mating, what's called pre-zygotic isolating mechanisms. In other words, the evolution of characteristics that prevent those mating mistakes in the first place. And that's fairly cheap. Making mistakes is costly. This is a lot cheaper than that. These are things called pre-zygotic isolating mechanisms. Behavioral differences, courtship differences, things like that. Differences in the time of year. So, if you go right to differences in mate recognition, if you go right to characteristics that have to do with mate choice, mate recognition, sexual selection, you can get very rapid reinforcement of differences and the completion of speciation. So how do you identify this? All right, again, you have to have sister species. They have to have some degree of geographical overlap. They have to be reproductively isolated from one another or be in the process of becoming isolated from each other on the basis of characteristics pertinent to reproduction. But there also must be evidence that they are secondarily overlapping geographically. There has to be evidence that they were initially isolated by a barrier which is no longer there. The ancestral species must have been subdivided by that barrier, or the species are old enough to have been affected by that barrier if you don't have direct evidence about the barrier at the time. Now, the interesting thing about this is that there's nothing that says that all the members of a given clade have to be formed by one kind of speciation. So it's possible that you can have a complicated set of speciation events that produces all the members of your clade. So let's say we have these 4 species, and here are the geographic distributions of those species. Now, what's going on there? Now you have A, okay, and B, C, D. Well, here's A, and this is B, C, D. You have B and C, D. That's this speciation. B and C, D. And then C and D, and D is completely contained within C. So this looks fairly simple. Vicariance, sympatric speciation. And it could be, depending on this, all these things I've been talking about. So the first assumption is that these distributions today are an accurate reflection of the ancestral distributions at the time of speciation. That's a tough one, especially the older the speciation event. The older the speciation event, the more you expect the current geographic distributions are not the ancestral ones. So that's what the ancestral distribution would have been like, something that included all of that. And the distribution, that was the speciation event there. That, you then have that in ancestral 2, that speciation event. Ancestor 3, and those two are sympatric speciation. So vicariance, sympatric speciation. If the current distributions accurately reflect what the distributions have been all along. The phylogenetic hypothesis is allopatric mode 1, allopatric mode 1, and then up there sympatric speciation. One there. Assumption number 2 in your study. Those are all the species that have ever been in the clade. You're not missing anything by extinction or sampling error. That's the complete story. What happens if we go out and find this later? Okay, now you have an additional speciation event. Now it turns out that C and D, which are sympatric, are not actually each other's closest relatives. E and D are actually each other's closest relatives, and they're not sympatric. These are sister species, and they're allopatric. And this ancestor plus that ancestor would not have been sympatric, they would have been parapatric. See? Now that's what that ancestors looks like. And then up here, that's allopatric. So now the story is different. You have vicariance, parapatric speciation, vicariance. So making sure that you have all of the members of your group is really important. It will really affect your kind of explanation. And those are two very different scenarios. And it's all changed by finding that additional species. So it turns out that things like basic taxonomic inventories are really important for studying evolutionary mechanisms. It's not just important that we know about all the species on this planet so that we can do what we can to adapt to the biodiversity crisis. Losing species, especially losing species before we know about them, is going to have an impact on the quality of our evolutionary explanations and our assessments of what kind of evolutionary mechanisms have been predominant. A world in which there has been a substantial amount of extinction of species with small geographic distributions and unusual ecologies is going to make us think that most of evolution has been simple vicariance. But that's going to be an artifact of two things: localized extinctions that we haven't documented, and poor taxonomic sampling. It's not going to be a reflection of the actual dynamics of the world. Now tomorrow, I'm going to end up talking about evolutionary radiations, which is mass speciation events. And it turns out that there is a way to compensate for those kinds of deficiencies in our database. But we're rapidly losing the ability to even do that. So there are a lot of reasons why we don't want a lot of diversity to disappear, at least not before we find out where it was. So here are some things that are important. Speciation produces sister species. You cannot study speciation one species at a time. Speciation is irreversible. Okay? Now you can pretend that population genetics will occasionally accidentally produce a speciation event as a byproduct. Or you can believe that species are caused by things. And you can study the difference. I mean, you can actually test that proposition. But you need to understand that speciation is forever. Speciation modes refer to factors that cause the initiation of speciation. And reconstructing that requires no kinds of dispersal, positive or negative, that has affected our ability to reconstruct ancestral geography, and there are no missing species in your phylogenetic tree. Allopatric speciation is initiated by the subdivision of an ancestral species. And there are two ways to do that. It's recognized by finding sister species with non-overlapping geographic distributions separated by a barrier, and there are different mechanisms that can complete allopatric speciation. So some people believe that vicariant speciation is speciation without selection. That's a very naive, superficial understanding of evolution. And for one thing, there's never a period of time when organisms do not interact with their surroundings. So there's never a point in time when selection doesn't exist. Whether selection affects the speciation event or not has to do with the completion of the divergence. In allopatric speciation, selection is not involved in initiating speciation, but that doesn't mean that it's not critical in understanding how speciation is completed. The majority of documented cases of speciation seem to be due to vicariance. But once again, we don't know how much of that is affected by sampling error and a lack of understanding about localized extinction. Non-allopatric speciation is initiated while populations of the ancestral species are maintaining some degree of gene flow. They're recognized by finding two sister species with overlapping geographic distributions. Primary overlapping distributions. Now, speciation is thought to be initiated and completed by selection in those modes of speciation. That's one of the reasons that it's attractive to some evolutionary biologists. They would like a simpler world where the same mechanism initiates and completes speciation, as opposed to allopatric speciation where one thing initiates and something else completes. And these are all the different ways we talked about before. These are really interesting, but there are very few well-documented cases in the natural world of these modes of speciation. So, here's the thing about species and speciation. If you look at some of the more standard textbooks about studying speciation, you'll be presented with a research program that's actually fairly simple. Now, you may be collecting a lot of data intensively, you may be sampling intensively, you may be doing a lot of sequencing, a lot of statistical analysis, or experimental studies of hybridization, and things like that. But, fundamentally, you're only doing one thing. You're collecting one kind of data and extrapolating to an entire process. And you generally end up starting with something like, I'm studying these two species because I'm interested in sympatric speciation. Now, what we're suggesting is that there is a more inclusive but more complicated way to go about this, where instead of starting with, I think this is sympatric speciation, let me see if I have any evidence to support it, you start from the other end of the process. You go, are these sister species? What's the geographic distribution? What's their biology? What's their reproductive biology? What's their ecology? What's their ability to move around? How much gene flow is there? Now that's a lot of information to collect for minimally two sister species and their closest relative to give you an outgroup perspective. So if you want to study how this one species originated, this is everything you're going to have to do. Now that's a lot more complicated, it's a lot more time consuming than most people who say they're studying speciation ever do. But if you follow that kind of protocol, at the end of the day, you end up with a lot of information. And you end up with a much more robust explanation of what actually happened in a particular case. Now again, this kind of research is very difficult for one person to do. Imagine trying to do all of that for a PhD dissertation. That's terrible. Now imagine a situation in which you have a group of cooperating specialists. You have a systemicist. You have a geologist. You have a reproductive person. You have a population geneticist. You have a molecular biologist. They all get together, and they and their students and their postdocs all work together. Now what happens? The whole process ramps up and goes really fast. Now you can do many groups quickly because you have a group of cooperating specialists. You have division of labor. Everybody gets something out of it, but nobody has to do all of it themselves. So one of the subtexts of historical ecology is, if you want to do this kind of research, if you believe that this is what is necessary to have a really robust evolutionary explanation, then the big problem for you is recognizing that the sociology of basic research in evolutionary biology is not adequate for evolutionary studies. When we thought that the story of evolution was very simple, individual people in individual labs could do the whole thing. If evolution has actually been this complex, then what we actually have to have is groups of cooperating specialists. And one of the best examples of that Deborah McClellan and I know is the group of people studying the evolution of insect-plant systems at Stockholm University. It is astonishing how much work they get done, how much they know. And it's all because they work together, they're good colleagues, they help each other out, they help each other's students out, they promote each other, they support each other, and in the end, everybody learns an enormous amount. I mean, on Thursday, when I talk about communities and coevolution, the chapter about coevolution and the nature of diversity, our last book, we used to refer to it as the Stockholm chapter, because the difference between that chapter in Phylogeny, Ecology, and Behavior in 1991 and the nature of diversity in 2002, everything changed. And that was all because every year for 10 years, Deborah and I would go and teach at Stockholm, and then we would go to a conference and listen to these folks and listen to their students. And I was like, And after a short 10 years, they were able to educate us well enough that we could produce a much better book chapter. But it can work. It's just that almost everything in the way academic science is set up selects against cooperation. And so that's part of the story here is if you're attracted to this kind of research, you have to be very, very careful and very diligent about what you do in terms of finding a research environment, finding a research group, or catalyzing a research group to do this kind of work. And I think that's one of the reasons that in the 1990s, everybody thought that they were all doing mapping traits on phylogenies and studying adaptation phylogenetically. And now there's very little of that being done, because all the easy stuff was done in the 1990s. Now we're left with the difficult stuff. It's interesting. Now I think this is much more interesting than just mapping a trait on a tree, but it's complicated. And if Deborah McLennan were here talking to you about how to integrate phylogenetic stuff with experimental studies and behavior, and how many thousands of hours of videotaping fish in the cold and the dark she's done, you would understand that this is complicated. But it's also very rich, it's very deep, and it's very profound, and it's very robust. And it tells us a lot more about the fundamental complexity of the evolutionary process than the way we've been doing things. So I'm not just saying, you should do it the hard way because you're younger and I'm older. Hopefully there's some sort of benefit in that cost. Okay, so we got questions, observations from either place? I'm letting you go early tonight. You're escaping an hour about 40 minutes earlier than yesterday.
I had some question. But you already asked.
Is the room in Moringa flooded? Did you guys have to come by canoe today? Okay, well, if there are no questions now, starting tomorrow, we will begin each one of these sessions. If there are questions about yesterday or today that you have for tomorrow, you folks here that are everybody in Moringa, let's start with that. If there's something that you think about, just write it down and ask me. I know that I get all really excited about this and sort of get going. Don't be afraid to stop me in the middle. Okay, I won't forget what I was talking about. That's a different joke. Okay, thank you very much, and we will see you. Some of you may want to come and talk to me this afternoon. I'll be up in Walter's area, Walter's lab, and otherwise we'll see you tomorrow.